Hey, all you cool cats and kittens, it's Mr. Foster here to teach you weeks 2 and 3 information. We're going to start off by talking about meiosis, which is something that y'all learned about last week for your first week of remote learning. It's also what we talked about a couple of weeks before we let out for spring break. So remember, meiosis exists for a couple of reasons. One of those reasons is to split the number of cells in half. You start with a diploid cell, which has two sets of chromosomes. See, we have two sets here. And you end with four daughter cells that are haploid or have one set of chromosomes, right? Half the number that you started off with. So that's one mechanism of meiosis. The other one is to mix all of those genes and chromosomes around so that each of the four cells looks just a little bit different, like the ones to the right here. So meiosis produces variation through independent assortment. Independent assortment basically talks about how each of the homologous chromosomes are going to be separated independently of each other. So if we look down here, you can see that each cell got one of each type of chromosome. So meiosis also produces variation through crossing over. So crossing over is whenever two chromosomes, two homologous chromosomes, so remember they share the same traits, okay, the same traits. So for example, if this chromosome had the gene for eye color, this one would also have the gene for eye color. Now, they trade pieces back and forth, and the resulting mixture is going to end up being four different chromosomes, right? Whereas you started off with just two different ones. An allele is a version of a gene. So, the brown eye gene or the blue eye gene, where they're both the gene for eye color, they're different versions of one another. Okay, so right here we have an allele as a version of a gene for a specific trait. So like I said, alleles are the different forms of a trait. There can be multiple alleles for each gene. Alleles are typically represented by a letter. And so for the purposes of what we're learning about, they're represented by a letter. And as you see here, we have a big B and a little b. That may be something that looks familiar. It's because a lot of y'all would have gone over this information in 7th grade. So alleles are the different forms of a trait. They can be dominant or they can be recessive. Dominant ones are represented by a big letter. Recessive ones are represented by a little letter. Because everyone has two copies of each gene, everybody has two alleles or two letters for every trait. This combination of letters is called your genotype. And that's what we're going to talk about next. So genotype is a combination of alleles in an individual represented by their two letters. So you get one letter from each parent, right? And your parents got each of their two letters from their parents or your grandparents. So the capital big letters, like I said, are called dominant, lowercase called recessive. They can be homozygous, which are the same, or heterozygous, which are different. So down here, we can look at examples of that. You could have homozygous dominant, which would be a big T and a big T, or you could have homozygous recessive, which would be a little t, little t. You could also have heterozygous, which is always going to be one big, one little, because like I said, hetero means different. So there's never going to be an instance where a heterozygous genotype will have two of the same letter. Okay, so moving on, let's talk about that dominant and recessive thing that I mentioned earlier. A dominant allele is expressed even if it is paired with a recessive allele. So for example, here, the dominant trait is going to show up because they're both big B's. Here, the dominant trait is still going to show up because the big B allele is stronger than the little b allele. Now, a recessive allele is only going to show up whenever it's paired with another recessive allele because it doesn't show up if it's being masked by a dominant allele. So this is the only instance where you will see a recessive gene actually showing up. So that means that you can have two different genotypes and they're still going to look the same. And that word is called phenotype, and that's what we're going to go over now. A phenotype is a physical trait that can be observed, or at least typically. An example would be the individual had brown eyes, right? Or had blue eyes. Blue eyes and brown eyes would be the two different phenotypes. But keep in mind that here with brown eyes, there's more than one genotype that can create that one phenotype. With blue eyes, the only genotype that can produce the phenotype of blue eyes is little b, because the big B is dominant. Brown eyes are dominant over blue eyes. So we're going to look at all of the different possibilities of what a baby could look like based off of the alleles that its parents have. So staying with the theme of eye color, we have the alleles, right? Allele for brown eyes, big B, allele for blue eyes, little b, and the possible genotypes that they could be within somebody would be big B, little b, or little b. And here's the phenotypes that each of those will produce. So like I said, homozygous dominant and heterozygous will both portray the dominant phenotype. The only one that will give you the recessive phenotype is if you have both recessive alleles. So we have another example here, this time with fruit flies, and the trait that we're looking at is wing texture. Okay, so we have our two different alleles. We have a big W, the dominant allele is for normal wings, and then we have the little w, a recessive allele, is for wrinkled wings. And so the only way that one of these fruit flies could be born with wrinkled wings is if they receive a little w from each parent, right? As long as they have one big W, they're going to have those normal wings. Now we can use this information that we just learned about, phenotypes, genotypes, homozygous, heterozygous, and apply that to something called a punnett square. A punnett square is basically a chart that can help you predict which genes could be given to an offspring from both parents. So it's a way to basically predict all of the different outcomes that could have arrived from independent assortment, which is what we talked about earlier. So we could take what we know now and apply it to eye color allele. So here's that same information now plotted into a punnett square. And I believe we have the steps for how to set up that punnett square on y'all's Google Classroom in your reference sheet. So make sure you're looking at that. So we have one parent up top, one parent on the side. We have that the mom is heterozygous, big B, little b, and that the father is heterozygous, so big B, little b. Now, if they both have a dominant allele, that means both parents are going to have brown eyes. And so if we look at the 4 possibilities for what their kids could turn out like, we see that there's a 3 out of 4 chance that the baby will have brown eyes, and a 1 out of 4 chance, or 25% chance, that the baby will have blue eyes. Keep in mind that there's 3 different genotypes here. We have big B, big B, big B, little b, and little b. And only one of these is going to show the recessive phenotype. Yet another example here, we have one parent who is homozygous recessive being crossed with a heterozygous parent. And so the genetic outcome is a 50% probability of being heterozygous with brown eyes and a 50% probability of being homozygous recessive with blue eyes.